Welcome everyone to our next TPD webinar. And today we have a very special webinar because it's very much topic focused, and we'll discuss this unique and very interesting ligase UBR5. And today we'll have 4 speakers from 3 different institutes. So we are very excited to do. Today we'll also try to take the questions at the end of all talks for both seminars. So it's my great pleasure to introduce 2 first speakers. Jonathan Tsai. Jonathan is the instructor in pathology at the Brigham and Women's Hospital and postdoctoral scholar in the laboratory of Ben Ebert at Dana-Farber. And Jake Aguirre from FMI. So Jake is the postdoc fellow at FMI in Basel, Switzerland, where he studied molecular mechanisms in protein degradation in Nicotoma lab. Thank you very much for accepting our invitation. We are very much looking for your seminar.
Great. All right, so hi everyone, thanks for the invitation. So Jake and I are excited to tell you a story we've been working on together, but a joint project between the Ebert and Tomei groups, looking at hormone receptor degradation. So nuclear hormone receptors are a family of related transcription factors that are involved in human health and disease. I think everyone here is familiar with a hormone receptor or 2. I think the 2 most prominent are the estrogen and androgen receptors that drive breast and prostate cancers, respectively. Other hormone receptors include LXRs that govern cholesterol metabolism. The retinoic acid receptors play a huge role in embryonic development and also drive a subset of leukemias. And there's just a battery of other hormone receptors that play super important roles in physiology. So hormone receptors as a whole mostly bind a ligand or a hormone, and that induces a structural change, allowing for the recruitment of a nuclear coactivator, leading to transcriptional activation. And then afterwards, a number of them are degraded by the proteasome. And so this has been documented first in the retinoic acid receptor alpha, or RARA, which is degraded by all-trans retinoic acid, or ATRA. The glucocorticoid receptor is degraded by corticosteroids like dexamethasone, and the estrogen receptor is degraded by estradiol. And so a key question in the field is, how are hormone receptors degraded? And this is important because hormone receptor degradation is very useful clinically. And so this is hallmarked by the use of the selective estrogen receptor degrader, or SERD, known as fulvestrant, which has been approved for the treatment of metastatic breast cancer. And so its target is ER. As I alluded to before, the retinoic acid receptor alpha, or RARA, is degraded by ATRA, and together with arsenic trioxide, those 2 represent a very robust non-chemotherapy treatment for acute promyelocytic leukemia with very, very high cure rates. And then hormone receptor degradation is just an intensely active area of investigation right now, as there are a number of PROTACs targeting ER and AR respectively for breast and prostate cancer. And finally, hormone receptors are structurally related because they're in the same family. They share this N-terminal domain, a DNA binding domain, and a ligand binding domain. And so our question or hypothesis was, is there a common degradation mechanism for hormone receptor regulation? And so to first get at this, we first interrogated RARA, as it's a prototypic hormone receptor, and it's altered in myeloid malignancies, so something our lab is familiar with. So the T1517 translocation fuses PML and RARA together to make this fusion protein that drives acute promyelocytic leukemia, or APML, by first forming a differentiation block in these precursors. PML-RARA is unique in that it's degraded by two different small molecules. So arsenic trioxide degrades PML, and ATRA actually activates and then degrades RARA, lifting that block in differentiation. And groups have previously shown that you need both RARA activation and degradation to get full APML treatment. And so to first verify this, we looked at ATRA-induced RARA degradation, first with whole proteomics. So we used an MB4 cell line, which is an APML line. And together with Catherine and Eric Fisher's group, we performed whole proteomics. And you can see that after 24 hours of 10 micromolar ATRA treatment, we get very robust and specific degradation of RARA and its heterodimeric binding partner, RXR alpha. Importantly, we showed that this degradation is proteasome dependent. So you can see that it's rescued by MG132, but it's importantly not netylation dependent. So it does not get rescued by netylation inhibitors. So to understand the E3 ligase machinery involved in degrading RARA, we first created a reporter cell line where we fused our protein of interest, so RARA, with EGFP and then an IRAS M-cherry. So by flow, we're able to very quickly ascertain the level of RARA protein by GFP and then normalize to transcriptional effects by M-cherry. So we created these reporter cell lines and then infected with a custom E3 ligase CRISPR library targeting every E1, E2, and E3 ligase and ubiquitinase in the genome. We let this cut for about a week, and then we treat for 24 hours with a ligand, in this case, ATRA, and we sort the top 5% of this population. And so this population represents cells that do not have deficient RARA degradation, presumably because they've received a guide for a crucial part of that machinery that's needed. And so this is the results of our RARA degradation screen. So this is actually two screens overlaid with each other. In red is ATRA treated and in gray is DMSO. And so you can see in the red, in the ATRA treatment, you have enrichment of a single E3 ligase, UBR5, as well as OTUD5, which is a deubiquitinase that stabilizes UBR5. And so again, on the x-axis, you have log2 fold change, and on the y-axis, you have a negative log of the p-value, suggesting that UBR5 may be regulating RARA degradation. And what was interesting is we repeated this screen for a number of hormone receptors and their ligands, so GR and dexamethasone, RXR and retinoic acid again, ER and estradiol, and LXR and a synthetic cholesterol. And you can see that UBR5 scores in every one of the ligand-dependent screens, but not in the control, suggesting that it degrades multiple nuclear hormone receptors. And so what is UBR5? So UBR5 is a HECT E3 ligase. So it's a family of very large ligases. And Jake will tell you it makes it very difficult to work with. And so as a consequence, not much was previously known about it until recently, where a paper a couple of years ago showed that it's recurrently mutated in mantle cell lymphomas. And you can see that these mutations cluster in the HEC domain, taking out the catalytic domain. Other studies have implicated it in MYC regulation, which the next talk will touch heavily on as well. And so to first validate this, we knocked down UBR5 using shRNAs and showed that this basically rescued ATRA-induced RARA degradation.
Yeah. Thanks, Jonathan. So I'll take over from there. This was actually about the part in the chronologically where we also got involved in the project. We were really excited by this finding of UBR5 being responsible for degradation of hormone receptors. And one of the first things that we wanted to do from the biochemistry side was try to determine if this interaction was direct or indirect. And we designed a very simple binding experiment to assay this. We were able to purify full-length human UBR5 from mammalian cells. We immobilized this and then assayed for binding of the retinoic acid receptors in the presence of various ligands or DMSO. And really strikingly, when we did this experiment, we saw that a direct association of UBR5 with the retinoic acid receptors, but this was only the case in the presence of agonist molecules. So 9-cis retinoic acid and all-trans retinoic acid, or an equimolar mixture of both, could stimulate complex formation, but an antagonist could not stimulate this and was comparable to DMSO. And interestingly, this was the case with the retinoic acid receptor alpha on its own, and a heterodimeric complex with its heterodimer partner RXR alpha seemed to be a potentially better substrate for UBR5. And this suggested to us that it's really only the agonist conformation of these retinoic acid receptors that is being bound by UBR5. And just to have a look at what this means on the molecular level, I want to look at some structures of these hormone receptors that have been solved by many other groups many years ago. What you can see, though, is that although these hormone receptors bind very structurally diverse ligands, as you can see here, they adopt very similar three-dimensional conformations in their ligand binding domains. And you see this on the overlay of the red and the pink structures, despite the fact that they bind very different ligands. And they have this C-terminal helix, which is also denoted helix 12. I've colored that here in blue. And this helix acts as a sensor for agonist ligands. And when an agonist molecule binds, this helix will undergo a rotation and uncover this binding site that is used for recruitment of various coactivator molecules. And just from looking at these two structures, we could already know that UBR5 is preferentially binding the structure here on the right. And that tells us already, or gave us a good idea where the binding site was, but we wanted to determine this experimentally. And we did this by doing mutational scanning. We first did an alanine and subsequently a glutamate mutational scan of nearly all surface residues across the ligand binding domain of RARA. And we could find that when we made mutations specifically in this cleft here that I've highlighted in blue, we could rescue ATRA-induced degradation, suggesting and confirming that this is the binding site of UBR5. We also showed this using our binding experiment, and we could show that a single valine substitution in RARA and RXR was able to abolish this interaction entirely. And so we now had quite a good idea how this interaction looked like from the perspective of RARA, but we didn't know how this looked like from the side of UBR5. As Jonathan said, at the time when we started this project, it was quite a poorly characterized protein. We had some isolated structures of small domains that were solved by crystallography and NMR, but this was it's a over 300 kilodalton protein. And we didn't know how the rest of this comes together. And because we had purified this protein already, we were able to look at it by negative stain and see these very large globular proteins suggestive of a globular quaternary structure. And with my Collis Lucas, we were able to do cryo-EM of this protein. And with some perseverance, we were able to determine the structure of this ubiquitin ligase, which I'll show here. And the first and most striking feature of this protein is that it is actually composed of 4 units of UBR5, which assemble into this homotetrameric ring shape. And this is a massive ring, almost 240 angstroms across. Here I highlight one of the monomers of UBR5. So you can see how it has a large dimerization interface here on the top, formed together by a helical bundle. And there's a second dimerization interface on the side, which holds basically together the 2 halves of the ring. If we want to have a closer look at the structure of one of these monomeric units for simplicity, we were able to solve this to higher resolution and could identify several new domains that had not previously been characterized. Together with my colleague Georg, we were able to build an atomic model for UBR5, which I show here, and we're able to determine structures of several new domains and features of this ligase that had not previously been determined. Just to briefly go through some of the major features, we found that at the N-terminus of this E3 ligase, there is an RCC1-like domain that is this large 7-bladed beta propeller, but it's broken up by 2 insertions. One of them is a ubiquitin binding domain, and the second one is the dimerization interface that I mentioned previously. There's a large helical scaffold in the middle, which is quite flexible and allows some bending of this protein in the context of the ring. And at the C-terminus, you have the HEC domain, which contains the catalytic moiety. And this HEC domain is also broken up into 2 sections by an intervening sequence by this Mademoiselle domain or MLLE domain, which is thought to bind substrates, although this hasn't exactly been shown in the structures. Sorry. And this structure is also remarkably consistent with also nice work from the labs of Hulin Lee, David Hasselbach, and Brenda Schulman, which published structures that are nicely in agreement with ours this year as well. I want to highlight the regions of this structure that we found were necessary for degradation of nuclear receptors. And we did this using our fluorescent reporter, which we treated with small guide RNAs to induce in-frame mutations and see how this affected the ability of the reporter to be degraded in response to ATRA. We identified several parts of this ligase that were necessary for degradation. The first, unsurprisingly, was the HEC domain that contains the catalytic cysteine. It was not exactly surprising, but what was surprising was this region that is separate in sequence, but in three-dimensional space folds back onto the catalytic HEC domain, and it wedges itself in between the N and the C lobe of the HEC domain, and therefore we called this region the wedge. And this seems to stabilize this predominant L conformation in at least the apo structure of this ligase, which likely has implications for catalysis. And I just point out that this is actually different than the only other HEC ligase structure that has been solved in its full-length form. That was done by Eric Fisher's group, where they identify this other conformation that resembles more of a T in their structure. At the N-terminus, we have a ubiquitin binding domain, which is able to bind ubiquitin and can affect chain linkage specificity of ligases. And we found that this was also important for degradation of RARA. So mutation or deletion of this domain led to greatly impaired ability of this enzyme to process ubiquitin chains and led to lower catalytic activity overall. And although we didn't see this UBA domain in our structure in three-dimensional space, we could see it in two-dimensional projections as this kind of blurry density that seemed to be moving around the ring in different 2D projections. And we could see that when we deleted this, we lost this density in 2D classification, suggesting that the UBA domains are in the center of the ring. And this would place them in proximity to the HEC domains, also facing inwards of the ring. And all of these moieties are quite flexible, and we think that this contributes to the ability of this ligase to process K48 linked ubiquitin chains extremely rapidly. And this was also shown very nicely in a paper by Laura Hale and Brenda Schulman, where they were actually able to trap catalytic intermediates and showed how the UBA domain directs K48 linkage specificity in this enzyme. Finally, we had a hit in the middle of the protein that was responsible for RARA degradation, and we think that this points to the substrate interaction site. So we weren't able to determine a structure by cryo-EM of this complex. However, by negative stain, we were able to quite easily see an additional density corresponding almost exactly to the size of a RARA RXR heterodimer that we would expect. And we think that this represents the tertiary complex. And the way that this interacts with RARA and RXR is quite fascinating, because as I mentioned before, this site that we identified as the binding site is also used by the various co-activator proteins and also co-repressor proteins. And this suggested to us that these processes likely can't happen at the same time and would probably be mutually exclusive. And this was something that was tested by Jonathan and the mass spec team in Boston, where they did time course IPMS experiments showing that following administration of ATRA, you get rapid disappearance of the co-repressor complex. This is replaced by the co-activator complex, and only at later time points do you see UBR5 taking over. And this was also shown by Western blots, that only UBR5 seems to appear at later time points in these experiments. And to show that this is directly competitive, we were also able to purify NCoA1 and show that it's able to outcompete the interaction with UBR5 in a dose-dependent manner, again confirming that these interactions are mutually exclusive. And this would suggest that that this switch from the activation to degradation at some point at a later time point happens, and this is allowing the eventual degradation of these nuclear receptors. And Jonathan will take back over.
Awesome, thanks. Right, so as Jake said, UBR5 binds to RARA and other hormone receptors, together in after NCOAs. And so as these transcription factors all exert their effects on chromatin, we wondered, where does the binding actually occur in vivo and in cells? And so to get at this, we performed ChIP-seq, where we pulled down either RARA or UBR5 with and without DMSO in these MB4 or APML lines again. And you can see here the predominant pattern is a loss of RARA peaks with ATRA treatment, which makes sense because it's being degraded and it's being lost. And what's fascinating is you can see that UBR5 co-localizes with RARA at all of these sites, suggesting that it does indeed bind to its targets on chromatin, potentially to degrade them off DNA. To really understand how this affects transcription, though, we performed RNA-seq with a RARA with an ATRA time course, so after 0, 8, 24, 48 hours after ATRA treatment, and we did this with and without UBR5 knockdown. And so as you can see here in the top left, we have a rapid induction, after 8 to 24 hours of ATRA-induced genes. And what's interesting is that when we knock down UBR5, we actually see a blunting of this effect, suggesting that UBR5 is actually needed for activation of the hormone receptor. And this was recapitulated when we treated with proteasome inhibitors, suggesting that it's actually the degradation of the receptor that's needed for hormone receptor activation. And so this is something that was a little surprising, and it's something we're actually actively working up now. And so just a quick summary, you have sequential binding on these hormone receptors, as Jake mentioned. So you have first binding of an NCOR or a co-repressor, then you have replacement with a co-activator after ligand binding, which is then replaced by an E3 ligase. And this all occurs on chromatin to regulate transcription. And so, the final part of the talk is whether UBR5 regulates other hormone receptors. And so I'll just show this screen again. As I mentioned before, we showed based on our screens that UBR5 probably regulates a number of other hormone receptors. And so we first decided to follow, secondly decided to follow up glucocorticoid receptor. And so GR, as I mentioned before, is degraded by dexamethasone. And so on the left, you can see an overlay again of two screens, dexamethasone in red over DMSO in gray. And you can see a very nice enrichment of UBR5 and OTUD5, even better than RARA in this case. When we knock down UBR5, you see that there's rescue of dexamethasone-induced GR degradation. When we mutate that same degron region, that hydrophobic cleft that Jake pointed out, we are able to prevent DEX-induced GR degradation as well, suggesting that they share a common degron. And then the ChIP-seq for GR is particularly elegant in that GR is a type 1 hormone receptor, which means it's bound at baseline to heat shock proteins in the cytoplasm. When it's activated by corticosteroid, it dimerizes and then translocates to the nucleus to then activate transcription. And then it's degraded. And so you can see that here, where we have nothing on DNA in time 0, in 15 minutes, and even in 5 minutes, we see rapid translocation in peaks with GR that's then lost after 24 hours. And what's cool is you can see UBR5 following GR around into the nucleus, onto the chromatin, suggesting that it's not only binding GR on DNA, but it's also, we can also recruit UBR5 to chromatin using a small molecule. And so the last hormone receptor we want to talk about very quickly is estrogen receptor, which is regulated in a UBR5 dependent manner by estradiol. So this is an IP pull down that Jake did recombinantly, and you can see that with estradiol, ER binds the UBR5 in the context of this ligand. But what was interesting is that he noticed that when you add fulvestrant, it actually kicks off UBR5 and it doesn't associate with UBR5 with that SERD. And so we wondered, we wondered what the other ligase was, what ER is binding to in the presence of fulvestrant. And so we performed the same CRISPR screens, and instead of UBR5, which was not enriched, we found enrichment of a different ligase, the sumo-dependent RNF111. And so we screened every SERD we could get our hands on because there's many undergoing development. And we found that we were able to basically stratify them based on chemistry and by E3 ligase employment. And so the basic side chain SERDs, including fulvestrant and the recently approved elastrant, all use RNF111, whereas the acrylic side chain SERDs use UBR5. And then these are PROTACs for controls using Cereblon and VHL respectively. We validated this by knocking down RNF111 with guides, showing that we could rescue fulvestrant and elastrant-induced ER degradation. And we showed using in vivo co-IPs that we could induce a fulvestrant-dependent, RNF and ER would bind in a fulvestrant-dependent manner. And so what was the most striking was that when we looked through the clinical trial data, we realized that all the basic or RNF111 using SERDs were either approved, so fulvestrant and elestrin have been approved, or are successfully going through phase 3 clinical trials, except for a couple of them that were discontinued in favor of these two. In contrast, all the UBR5 using SERDs were all terminated early and discontinued in favor of one of these RNF111 SERDs as well. And we think that's because these UBR5 using SERDs also engage NCOAs right before, leading to agonism of this signaling pathway, whereas RNF111 perhaps does not. And so it'll be interesting to see how the PROTACs do as well. And so quick summary, so we're a little bit over time. So hormone receptors undergo ligand-induced degradation. UBR5 is a general regulator of nuclear hormone receptor stability. It importantly has ligand-dependent binding, and this occurs in this conserved hydrophobic cleft, allowing it to bind a number of hormone receptors. It exists as this tetrameric E3 ligase. There's multiple domains like the HEC domain, which is needed for degradation. The UBA domain, as Jake mentioned, needed for chain elongation, and the hinge is needed for binding. Importantly, it competes with coactivators for binding and binds the hormone receptors sequentially. And all of this occurs on chromatin to activate transcription. And so this is really just a joint effort between two labs, Nico's lab and Ben's lab. You can see on the left to the right. And you can notice that there is no group picture of both our labs together. And so we're currently trying to pressure Nico into funding a trip for everyone in the Ebert lab to visit in Switzerland. Thank you.
Thanks for the great talk, Jonathan and Jake. And for the sake of time, we will move on to our second speaker team right away. So it's my great pleasure to introduce Dr. Kevin Mark and PhD candidate Durga Kolla from both the Miescher lab at UC Berkeley. Kevin received his PhD from UCSF, and he has focused on determining the role of protein ubiquitination in different eukaryotic systems to understand how ubiquitin-mediated protein quality control regulates gene expression during development. And Durga is a PhD candidate in Miescher's lab, and she's interested in molecular mechanisms underlying protein degradation pathways in development and disease, and currently focuses on quality control of protein complex composition in transcription. And thank Kevin and Durga for joining us today. And now the floor is yours.
Awesome, thank you.
Thank you.
Just give us 1 second to pull our slides up. Okay, is everyone able to see the slides?
Yeah, looking good.
Okay, awesome. Okay, so hello everyone. I guess my introduction has already been given, but I'm Durga and I'm a graduate student in Michael Rappa's lab, and I'm really excited to share work with you all today. So I really appreciate your time and thank you for being here.
I'm Kevin. I'm a postdoctoral fellow in the Rappa lab, and I want to again thank the organizers for giving us the opportunity to present our work. And we're happy to tell you about how we've identified the UBR5 as an orphan quality control ligase and how we think that this mechanism is used to promote gene expression.
So I'm going to start off by presenting the first half of our story. So many of us know that our body contains hundreds of protein complexes that consist of many different subunits that need to accurately come together at the right place and at the right time during development. And we know that nascent proteins interact with their binding partners co-translationally and can be assisted by chaperones to assemble accurate complexes. However, errors can occur in this process, sometimes due to protein misfolding or mispaired complex partners that can lead to flawed or excess assembly of complexes. And this can really be detrimental to key signaling events and many different developmental processes. But what's really amazing about our cells is that we have strategies to correct these errors. And one of these strategies, as probably everyone's favorite strategy, is the ubiquitin proteasome system through different quality control mechanisms. So this can prevent, correct, and degrade aberrant complex components. So one major class of multi-subunit complexes that's really essential for development are transcription factors. So here I'm showing, for example, a BTB family transcription factor must form homodimers to go on to bind to DNA and activate gene expression, whereas aberrant formation of BTB heterodimers can lead to improper nervous system development. And work from our lab has discovered a surveillance pathway for these BTB complex assembly called dimerization quality control. And here you can see E3 ligase, FBXL17, is able to recognize and target inactive BTB heterodimers for degradation, thus only allowing active homodimers to be maintained for their proper function. So really, apart from these dimerization motifs, we know that transcription factors have many intrinsically disordered regions that are able to recruit gene expression regulators into these condensates called transcriptional hubs. And while gene expression machinery is about 1,000-fold higher in these condensates than the nucleoplasm, these hubs still have to remain highly dynamic. And we know that even small changes in protein levels can alter the function of these hubs and lead to dysregulation of gene expression in many different disease types. And one way in which the cell can regulate protein networks and transcriptional hubs is through the use of the UPS. And while we kind of understand how proteostasis is maintained in the cytoplasm, transcription factor regulation in these nuclear condensates is much more complicated due to these transcription factors having to interact with many different multi-subunit complexes. So really, how these cells are able to ensure proper complex formation and function within these transcriptional hubs is not very well understood. So today, Kevin and I are really excited to share with you a new regulatory role for orphan quality control and establishing interaction within transcription factor networks in order to allow efficient and adaptable gene expression programs. So recently, our lab has identified regulators of stem cell self-renewal in an shRNA screen that I'm showing here in this figure. And you can see in this bottom left quadrant, hopefully you can see my mouse, my pointer, you can see these are all factors showing, that are critical for preserving pluripotency. And one of the factors that stood out to us is this HECT-E3 ligase, EBR5, which is really intriguing because we know that it's highly conserved, and loss of UBR5 is embryonic lethal in mice. And we also know that UBR5 mutations can be attributed to 18% of mantle cell lymphoma cases. And not only that, but we know that UBR5 is amplified in some cancers like breast and ovarian cancers in order to support tumor growth and metastasis. So really understanding the mechanisms for how UBR5 can be not only a regulator of stem cell identity, but also act as a tumor suppressor or oncogene remained unclear. So in order to begin dissecting this question, we started off by performing IP-mass spec, looking for interactors of endogenous UBR5. So here you have two IP-mass specs in 293 T cells and HeLa cells. And what I want to highlight from this scatter plot is that UBR5 is able to associate with three different transcriptional complexes, one being the NO80 chromatin remodeling complex, which is really important for maintaining an open chromatin structure at promoters and enhancers. We also see that UBR5 binds to two elongation factors, SPT4 and 5, as well as components of the mitotic checkpoint complex, and PLK1 and KIF2A, which all regulate the NFA's promoting complex to promote transcription initiation. So what's really striking about all these UBR5 interactors is that many of them engage with WDR5, and WDR5 is a methyl histone reader important for recruiting c-Myc to chromatin. And we also know that many of these proteins can bind directly to c-Myc, which I'm sure many of you in the audience know is a master transcription factor that plays a pretty essential role for establishing establishing stem cell identity. And we know that its c-Myc is frequently mutated in many, many cancers. So just as a quick thing, I wanted to point out the validation of this mass spec through small scale IP validation. And here you can really see, along with c-Myc and all the other transcriptional complex components from the mass spec, you can see pretty robust binding to UBR5. So now that I've just showed you that UBR5 has all of these different interactors, we wanted to understand, is UBR5 able to induce the turnover of its binding partners? And in order to do that, we first began by creating a library of transcription factor reporters. So very similar to the previous group, we have our proteins of interest tagged to GFP and IRS and mCherry serving as an internal control. And we've cloned about 100 or so of these transcription factor reporters. And then using siRNA, we can knock down UBR5. And then using subsequent FACS analysis, we can read out our reporter stability using the GFP and mCherry ratio to determine whether we have a UBR5 substrate or not. So essentially what you would see is with UBR5 knockdown, you would expect to see a stabilization or accumulation of your protein compared to control levels. So essentially a higher ratio value. So I'm going to cut to the punchline before describing this figure. What was really exciting, excuse me, to us is our screen not only revealed one substrate of UBR5, but 21 candidate substrates. And we really felt like we won the lottery here. So here I'm showing you the waterfall plot. And you can see on the x-axis all the different candidate substrates that we tested. And on the y-axis, you can see the median fold change of the GFP and mCherry ratios upon UBR5 depletion. And what hopefully you can appreciate is that you can see many of these regulators were hits found in the IPMS spec. So you have these chromatin remodelers, you have major transcription factors, you have components of the MCC, the two elongation factors, and so on. So here I just want to show you one example. We did throughout the paper focus a lot on some of our top hits, one of them being c-Myc. So just as controls, here you can see in all the red histograms control levels of c-Myc, and upon different upon the use of different inhibitors, for example, the E1 enzyme inhibitor, P97, and proteasome inhibition, you can see accumulation of c-Myc, telling us that it is part of this ubiquitin proteasome pathway, whereas with vancomycin A treatment, which is an autophagy inhibitor, you no longer see an accumulation of c-Myc. And we've tested this for all top 21 UBR5 substrates, and you see the exact same results. So here I'm showing you one of our, from the screen, you just see a dot, but here's the histogram image. So using CRISPR, we were able to make a UBR5 knockout line in HeLa cells, and you can see an accumulation of c-Myc compared to control levels in wild type HeLa cells in red. And additionally, using CRISPR, we were able to generate catalytically dead mutant cell lines of UBR5, which also lead to a stabilization of c-Myc. So far I've shown you that endogenous UBR5 can bind and destabilize multiple candidate transcriptional substrates within a network that's really centered around major transcription factor c-Myc. So next we wanted to know and test whether UBR5 is directly targeting its substrates via ubiquitin chains, leading to their subsequent degradation. So in order to do this, we purified endogenous UBR5 from HeLa cells and incubated it with E2 UBE2D3 and our candidate substrates that were produced via in vitro transcription and translation. So I'm showing you here c-Myc. So you can see that UBR5 is able to robustly ubiquitinate c-Myc. And very similarly, this is true for N080 complex members MCRS1 and REVBL2, as well as elongation factor SPT5 and MCC component CDC20. And here I'm showing you our top substrate MCRS1. So we just wanted to validate that UBR5 catalytic activity through the HECT and the UBA domains is required for ubiquitination of its target substrates. So upon mutation, you no longer see ubiquitination. And then what's cool is using ubiquitin lysine mutants, we can test which linkage type UBR5 prefers by visualizing a reduction in chain formation indicated by no schmear, essentially. So here you can see that UBR5 strongly prefers K48 to modify MCRS1 with K48 linkage types or K63 linkages. And we do have further experiments that show that it really is specific mostly to K48 linkages only. And I'm just going to take a break for a second and hand it over to Kevin to continue with presenting our work.
Thanks, Durga. So now that we had established that transcription factors and transcriptional regulators were major substrates of this ligase, we then asked, how does the global gene expression profile change in the absence of UBR5? And to do this, we performed RNA-seq from UBR5 knockout cells. And so on the right side, these are genes that are upregulated upon UBR5 loss, and on the left are those that are downregulated by loss of the C3. And what is immediately apparent is that UBR5 is not simply a negative regulator of gene expression. You don't see just genes appearing on the right side. In fact, there's just as many, if not more, on the left. And so UBR5 is important for sustaining gene expression. And this is true certainly for c-Myc targets, which we knew was a direct substrate of the C3. And so we first used quantitative PCR to look at the strongest differentially expressed genes, first starting out with the ones that are repressed by UBR5. And what I want you to notice is that cyclin D1 is a gene that's one of these such genes. And this is really interesting because cyclin D1 is often misregulated in mantle cell lymphoma. As Durga had mentioned, this type of cancer frequently contains truncations in the HEP domain and therefore loss of function. In addition, we identified RUNX1. As many of you in the audience know, this is a transcription factor that's commonly aberrant in hematologic malignancies. Now, again, what was really striking was that many of the genes that were differentially regulated were lost. Their expression relied on UBR5. And so again, here, many of these included MYC target genes, developmental factors. And so this is really perplexing because we knew that MYC was a target. It's degraded by UBR5, yet somehow the turnover of MYC was needed for the expression, at least for a large subset of target genes. And so to resolve this seemingly paradoxical situation, we first had to ask, how does UBR5 recognize its substrates? And we chose MYC as the prototypical example. I'm showing here the domain structure. And we did this using flow cytometry. We generated mutant constructs of MYC and then asked, how has their stability changed? And so we first looked at the N-terminal transcriptional activation domain. This is a common site of regulation for MYC. You can see that there are several MYC boxes that mediate protein-protein interactions. You also have a dagron in this area for FBXW7, another well-known MYC ligase. But when we removed the TAD, we didn't see any change in the ability of UBR5 to degrade this. It's actually when we got rid of the very well-conserved DNA binding domain at the C-terminus that you see a strong stabilization of the red curve has now shifted to the right compared to wild type. And importantly, when you get rid of UBR5, you see no further gain in stabilization, suggesting this construct is now resistant to turnover. Using further mutagenesis, we were able to create point mutations to narrow down two independent dagrons, one site in the basic region and one site in the leucine zipper. And this was able to recapitulate the UBR5 loss of function. Now, there was a crystal structure of MYC and MAX, the complex on DNA, solved by Nair and Berkeley in the early 2000s. And when we superimposed our dagrons on this structure, what was readily apparent was that they overlap with functional sites. So the first dagron is a basic patch that interacts with the negatively charged phosphate backbone of DNA. And the second dagron is buried within the MYC-MAX leucine zipper, the interaction region. And so the implication of this was that if MAX were present and recruits MYC to DNA, the dagrons should not be available for recognition. And to test this idea, we modulated the level of MAX and asked how MYC turnover is affected. So we first, here in orange, overexpress MAX and asked what happens to MYC stability. And you can see that there's a shift to the right, so this construct is stable. And when you get rid of UBR5 on top of that, you see no gain in stability. In other words, MAX overexpression blocks the turnover. At the endogenous level, we were able to deplete MAX, and you see a very strong loss of MYC, likely due to destabilization. And when you co-deplete UBR5 on top of this, you're able to rescue and regain all of that MYC protein. And so what both of these experiments suggested was that UBR5 is targeting MYC, but it's not targeting it in the context of the MAX-MYC dimer. That rather, when you render MYC an orphan, that's at the point at which it can be degraded. We were able to extrapolate this to other substrates that we had found. Here I'm showing you one example. The leucine zipper transcription factor MAF-F has a binding interacting partner of BOC2. Again, when you overexpress BOC2, MAF-F is stabilized, and this protects it from UBR5 dependent turnover. Again, you deplete BOC2, you destabilize this transcription factor in cells, and when you co-deplete UBR5, you can restore the protein. Now, we wanted to get an even further detailed understanding of how orphan quality control is specified by this ligase. And here we turn to Jake and his collaborators at Nico Tomei's lab. And again, they had a beautiful structure of cryo-EM structure of UBR5, and they were able to determine the MCRS1 substrate bound cryo-EM density. And so here you can see in this region is where MCRS1 binds. And through a combination of biochemical purified protein binding assays, as well as cross-link mass spectrometry, they were able to position and orient the FHA domain, which a region we knew that Degradation lay in, to very close to the catalytic HEC domain. And so here in black, you can see that this is the Degradation that we identified, and that it's very nearby the catalytic cysteine. And what's really cool about this is if you notice, MCRS1's domain is kind of pinned between, kind of sandwiched between two regions of the ligase, the tandem edge, as well as the HECT. And we think that this is what specifies the orphan protein, specifies the targeting of only orphan substrates. In this position, MCRS1 will not be able to interact with binding partners. Now, quality control is often thought of as targeting non-functional subunits that have either aggregated or misfolded and are detrimental to cells. And if this were the case, then if you get rid of UBR5, you're accumulating a non-functional population of MYC that should not be able to assemble into MYC-MAX dimers. On the other hand, if UBR5 is indeed going after monomeric subunits of MYC, then we can imagine changes in UBR5 would lead to effects at the mature dimeric MYC-MAX factor. And so to distinguish between these two models, we had to come up with a way to measure the stability and abundance of the dimeric MYC-MAX state. And so to do this, we use cyclohexamide chases. So I first want to bring your attention to the bottom panel. This is a Western blot of wild type versus UBR5 knockout c-MYC levels. And again, consistent with the flow cytometry, we see that endogenous MYC is more stable in UBR5's absence. Now, when we purified MYC-MAX dimeric complexes from these cells, we saw that, again, you have, with the accumulation of c-MYC in the total pool, many of these monomers, many of the c-MYC protein can still bind to MAX, and therefore you're getting a gain in the MYC-MAX dimer complexes, arguing for the latter model, that these UBR5 is targeting MYC protein that's competent to refold and assemble dimer complexes. Now, the constant turnover of transcription factors by UBR5 implies that cells may need to resupply, constantly resynthesize these factors. And so to test whether this was true, we turned to ribosome profiling data as a proxy of translation and to RNA-seq data as a measure of overall transcription. And what's very clear is that MYC is synthesized, produced more than in abundance compared to its stabilizing partner MAX. And again, this is true for MCRS1 and CCDC85B. And the implication from this was that if anything affects the rate of translation of these factors, perhaps that would affect the transcriptional complex formation and gene expression output. And so we knew from the work of others that the one way that cells can rapidly inhibit translation is through stress responses. And we treated cells with oligomycin antimycin A to activate the integrated stress response, and this rapidly blocks protein synthesis. You can see under these conditions that MYC-MAX dimers quickly come apart, whereas in UBR5 knockout cells, this happens at a much slower rate. And this has consequences on the gene expression response. You can see that under these same stress treatments, that under wild type conditions, certain genes are rapidly turned off. However, in the absence of UBR5, First of all, they are no longer dynamic, and second of all, they're completely blunted in expression. And what's really cool about this is that not only do we think that UBR5 regulates the dynamics of protein complexes, but that this further has impact on the complex interactions. So what I mean by that is if you take a look at MCRS1 in this IP, you can see that it tightly associates with transcription elongation factors. But when you get rid of UBR5, you see a switch, a loss of elongation factor binding, and a retention of upstream initiation factors, such as transcription factors like c-Myc and INO80 chromatin remodeler subunits. And so there's a failure to transition from one mode of transcription, one step of transcription to the next. And so I want to stop there and summarize our findings that we've shown that UBR5 targets transcription factors, many of which occur in dimeric complexes or multimeric complexes. There's an unstable component to these, and there's stabilizing members, and that these must interact, bind to be recruited to DNA to recruit coactivators and produce gene expression responses. But after these are used, they must come apart. And at that point, once they disassemble into individual subunits, the transcription factor, such as MYC, is degraded through its orphan interface, releasing the stabilizing partner to then rejoin a newly synthesized subunit, forming a dimer, which goes off to either reactivate that same locus to sustain transcription or to go to other sites on DNA. And we know that stress impinges upon this circuit by preventing the new synthesis of transcription factors. And coupled with the degradation, you rapidly clear DNA of these gene expression machinery. And so in thinking of how we can imagine using this to advantage for targeted protein degradation, hopefully we've demonstrated to you that UBR5 targets many transcription factors and transcriptional regulators, and it does this at the endogenous level. It naturally recognizes these machinery, as many of you know are very important substrates of TPD. Secondly, UBR5 is highly expressed in many cell types and tissues of the body, and therefore it's available for use as a TPD degrader. As Durga mentioned, and Jonathan mentioned, UBR5 is commonly amplified in cancers, and therefore it would be advantageous to use in these as a treatment for these diseases. And lastly, if we can imagine shifting the complex equilibrium, for example, there have been small molecules that have disrupted the Myc-Max dimer interface. If we can break apart these complexes, these molecules might therapeutically synergize with the effect of a UBR5 degrader. And I encourage you to further look at a review that was recently published by Asad Terboy and Daniels that covers these topics and more. So with that, I'd like to stop and acknowledge everyone who contributed to this project. This is a wonderful team effort by members within the ROPA lab, as well as a fantastic collaboration with Jake and colleagues and Nico's lab at EPFL.